Hello, I'm Dr. Yonathan Goldtzvik. This is an introductory training on similarity metrics and confidence scores that are commonly used in protein structure prediction. In this training, I'll give you an overview of different ways with which we can compare protein structures or determine the quality of predicted protein models. I'll also discuss the strengths and weaknesses of different metrics used to determine model quality and when it is appropriate to apply these metrics. I will first give an introduction on methods to compare protein structures. I'll then discuss how these methods were used to develop confidence scores that determine the quality of predicted models. I will also discuss particular metrics that are suitable for the analysis of predicted models of protein complexes as opposed to individual proteins. Finally, I will discuss the limitations of these methods and also how to interpret them. Let us start with protein similarity. Why might we want to calculate protein similarity? Proteins are chains that usually have a well-defined structure. And because of this regular and well-defined structure, it is possible to structurally compare different proteins to see if their shape is similar. One reason we might want to do this is that we may be interested in categorizing proteins based on their structure and get a sense of protein structural diversity. There have been several major and successful efforts to categorize all known protein structures. Due to these efforts, we were able to identify different protein structural families, and this was possible due to our ability to quantify protein structural similarity. Another important reason for why we might want to calculate protein similarity is if we want to compare predicted models to experimentally determined structures, in order to see whether our algorithms produce reliable predictions. Imagine that we have an algorithm, AlphaFold for example, that generates structural predictions. We want to see if these predictions are actually close to what we observe in experiment. For that, we need some kind of a metric. To summarize, the reason we need similarity scores is to have a quantitative and consistent way of comparing protein structures that can be used to detect structurally similar proteins or to assess prediction models. Hopefully, I convinced you at this point that it is important to calculate protein similarity. But how is it actually calculated? Let's discuss some ways in which this is done. Let's start with a naive approach to protein structure comparison. Imagine we have two protein structures that at least appear at a glance to be structurally similar. What we really want to do is calculate the differences between these structures. In order to calculate these differences, the first thing we need to do is to align these two structures somehow, and then measure in some quantitative way what these differences are. The basic approach that has been used to do this is something called RMSD, which is short for root mean square deviation. So how do we calculate RMSD? Imagine we start with two spatially aligned proteins. What do I mean by that? If we are comparing two proteins whose length is identical, there is a one-to-one correspondence between the residues of both proteins. An alignment in space simply means that we position the two proteins in space in such a way as to optimize the agreement between the two structures. There are different algorithms that can achieve this. In a case in which we're comparing two proteins whose lengths are different, we need to somehow decide which residues in one of the proteins correspond to which residues in the second protein. This is usually done in such a way as to optimize the alignment. Once the two structures are aligned, we can begin our calculation. If both protein structures are highly similar, the positions of each residue in space will be similar in both models. In contrast, if there are differences between the two structures, the same residue might have very different positions in each of the structures. Let di be the distance between the position of residue I in structure 1 and structure 2. RMSD is simply the square root of the mean of the squared distances, as can be seen in this formula. The more similar two protein structures are, the smaller the RMSD score. The more different the structures are, the larger the RMSD value. Generally, the convention is that RMSD values of 2 angstroms or less indicate a high level of similarity between two protein structures. One well-known and important issue with RMSD is that it is affected by the length of the proteins that we're comparing. Essentially, when comparing two short proteins, we're calculating fewer distances than what we would be calculating for two long proteins. Because there are always some differences between protein structures, longer proteins tend to have larger RMSD values associated with them. A particularly successful similarity score that circumvents this issue is the template modeling score, or TM score for short. This is a widely used similarity score in structural bioinformatics. In terms of how it's calculated, without going into detail, the formula for the TM score, which you can see here, includes a term d0 that depends on the length of the target protein. This term corrects for the length of the protein, making the TM score protein length independent. In addition to not being dependent on protein length, there are other important differences between the TM score and RMSD. The TM score runs from 0 to 1, with 1 indicating perfect structural similarity between two proteins, and 0 indicating no similarity at all. Additionally, the TM score considers multiple structural alignments as input. It then uses the alignment which results in the highest TM score, with only aligned residues included in the final calculation. Despite its success, even the TM score has its limitations. Consider the following scenario. Imagine two proteins, each consisting of two protein domains. These proteins are almost identical to each other in structure. The only difference is that the orientation between the two domains is different in each one of these proteins. You could think of one of the residues in the connecting linker between the domains as a sort of a hinge. Imagine calculating RMSD and TM scores in this scenario. In order to calculate either score, we need to align the proteins. But there's always going to be a disagreement. At best, we can always align only one domain at a time, with the other domain having a shift. Because of this, calculating the TM score or RMSD is always going to be affected by domain orientation, and aligning such structures is never a trivial matter. In order to circumvent this issue, we need a local metric that is not sensitive to domain orientation. The Local Distance Difference Test, or LDDT for short, is a similarity score that is particularly suitable in this situation. It measures local similarity and is indifferent to domain orientation. Furthermore, it's alignment independent, meaning you don't need to obtain a structural alignment in order to calculate it. So how is LDDT calculated? Similarly to other similarity scores, we need to obtain a one-to-one correspondence between residues. However, we don't need to align them in space. We first choose a cutoff distance. For each residue, we identify all of its neighboring residues that are within the cutoff distance in both the structures we're comparing. We then calculate the distances to these neighbors in each of the structures and determine what percentage of these distances remains unchanged using varying thresholds. This percentage is the LDDT score for that residue, and it runs between 0 and 100. The LDDT score can be calculated for individual residues or averaged over the entire protein. Because LDDT can be applied at the residue level, it is possible to see within each of the two proteins we're comparing which regions are the most similar. In terms of value ranges, values of 80 and above indicate high levels of similarity, whereas lower values indicate some similarity, and values below 50 indicate poor similarity. As I mentioned earlier, LDDT is a local score, meaning that information about the interdomain orientation is lost, and this is by design. However, we may be interested in recovering this information and may want to use a supplemental similarity score that focuses on interdomain orientation. A similarity score that was particularly designed to capture interdomain orientation is the aligned error. The aligned error is calculated between pairs of residues. Let's see how this is done. We first choose two residues in one of the proteins. Let's call these residues I and J. We now look at the corresponding residues I and J in the second protein. We use residue I to align both proteins. What do I mean by that? We align both proteins in such a way as to optimize the overlap between all the atoms belonging to residue I in both proteins. Once we aligned both proteins around residue I, we measure the distance between the C-alpha carbon corresponding to residue J in protein 1 and its position in protein 2. This distance is the aligned error score for this residue pair. In cases in which the orientation between the two domains remains relatively similar in both proteins we're comparing, we would expect small distances and therefore small aligned error values. In a case in which the orientation is quite different, we would observe large distances and therefore large aligned error values. Because the aligned error is calculated for each pair of residues I and J, we obtain an aligned error matrix. Here you can see a heat map visualization of such a matrix corresponding to a multi-domain protein. Darker regions in the heat map indicate low aligned error values, and brighter regions indicate high aligned error values. In this example, the dark regions clearly correspond to protein domains whose structure is similar in both proteins. In contrast, the regions in the matrix corresponding to interdomain distances appear brighter, indicating significant differences in the interdomain orientations. An important thing to point out is the aligned error value for residues I and J is not equal to its value for residues J and I. Switching the order changes the aligned error value. The reason for this is simple. If we align the residues J instead of residues I, we get different distances. As a result, the aligned error matrix is not symmetric, even though it might appear so at a first glance. Now that we've covered similarity scores, we can move on to discuss how we assess the quality of protein structure predictions. Prediction scores are similar to similarity scores, but while similarity scores measure just that, structural similarity, prediction scores measure our confidence in our predictions. Many prediction scores are based on similarity scores. Some examples are pLDDT, which stands for predicted LDDT, predicted align error, or PAE, and predicted TM score, or pTM. These scores correlate with prediction quality. pLDDT is perhaps the best known prediction confidence score. It follows similar principles to LDDT and can be calculated for each residue. It is the confidence score of choice for AlphaFold. Similarly to LDDT, its values range from 0 to 100. But unlike LDDT, it does not indicate similarity between two proteins. Instead, it has been shown that pLDDT correlates with the LDDT similarity score between predicted structures and their experimentally determined counterparts. It is important to note that because these scores are only correlated, a poor prediction could have a high pLDDT score, and similarly, a great prediction could have a low pLDDT score. In general, scores of 90 or higher indicate a high confidence in the model. A score of 70 or higher suggests that the model is mostly correct and that we should be reasonably confident in it. Scores below 70 are considered low confidence, and we should not trust the model in these cases. Earlier, we showed how the aligned error indicates similarity in terms of interdomain orientation. In an analogous way, the predicted aligned error, or PAE, indicates how confident is our prediction in terms of interdomain orientation. PAE scores are calculated in the following way. For each pair of residues I and J, a distribution of aligned error values is generated. Then, the PAE score is simply the weighted mean over this distribution. As mentioned earlier, PAE scores are assigned to pairs of residues, but we might be interested in having a global PAE score that indicates our confidence in interdomain orientation in general. To do this, we can combine the PAE score together with the TM score to generate such a global metric. The original TM score consists of a sum over distances. In a similar way, instead of considering distances, we can simply consider the PAE scores between pairs of residues. So a naive approach would be to replace the distances with those PAE scores, as you see here. In the case of TM scores, we look at different alignments. For each alignment, we calculate the sums of terms that depend on the distances over all the different residues, and at the end we choose the one that gives us the highest TM score. In the case of pTM, we looked at different residues I. For each residue I, we look at the sum of these terms of the PAE scores between residues I and J, and we sum it over residues J. We then choose the residue I that gives us the maximum pTM score. When considering the term in the sum, we can think of it as a specific pTM score that is associated with residues I and J, and we can call this pTM I and J. We can then think of the pTM score as choosing the residue I that maximizes the value of the mean over J of these pTM IJ terms. It is important to note that there are subtle differences between the naive approach that I just presented and how these pTM scores are calculated in practice. These, however, are beyond the scope of this training. Nevertheless, the essence of how it's calculated remains the same. Up until now, we discussed scores that help us assess the quality of predicted protein structures. We're now going to move on to assessing the quality of predicted structures of protein complexes. For simplicity, let's consider a protein complex that consists only of two proteins. Similarly to how we calculated the PAE matrix for a single protein, we can calculate the PAE matrix for the protein complex. We can then use PAE to assess the confidence in the interprotein orientation in our model, in a similar way to how we used it to assess our confidence in the interdomain orientation in a single protein. In theory, we could calculate the pTM score for the entire complex. We would just use the entire PAE matrix. In practice, we may be interested only in the regions in the PAE matrix that correspond to the interface between the two proteins in the complex. Low PAE values in these regions of the matrix would indicate high levels of confidence in the placement of these two proteins relative to each other in our model. High values of PAE would indicate low confidence. Instead of calculating the pTM score over the entire matrix, we can calculate it only for the interface regions between the two proteins. We call this new score ipTM, or Interface Predicted TM Score. Because the PAE matrix is not symmetric, it is important to calculate the ipTM score over both interface regions and then choose the one that gives us the maximum value, in a similar way to how we've calculated the TM scores and pTM scores. Now that we've covered the most important and widely used confidence scores in protein structure prediction, it is important to discuss how to interpret these scores and their limitations. As discussed earlier, pLDDT is a good estimate of prediction quality in general. However, there are some caveats. It is correlated with the actual LDDT score between the predicted structure and the experimental structure if you have one. However, it is not exactly the same thing. You could have high pLDDT scores when the actual LDDT is low, and vice versa. Therefore, we should think about it more as how confident is AlphaFold in its prediction. It has been suggested that low pLDDT scores are associated with intrinsically disordered regions in a protein. However, this association should be interpreted with caution. pLDDT scores are based primarily on the amounts of evolutionary data available to AlphaFold when it makes its prediction. Low pLDDT scores could be an indication of an intrinsically disordered region, but they could also be an indication of not having enough data in order to make a confident prediction about a protein structure. Those things should be kept in mind. An important weakness that has been identified for TM scores, and in turn for ipTM scores, is that they are affected by the difference in length between the target protein and the template protein. Specifically for ipTM, discrepancies in regions that are not directly involved in an interaction affect ipTM scores negatively. Therefore, we might find ourselves in a situation in which a low ipTM score is not actually a correct indication of how confident we should be in our model. It is important to note that there are alternative scores that do circumvent this issue. These are beyond the scope of our training today. To summarize, pLDDT scores are useful indicators of how confident we should be in a protein model prediction. When we want to assess our confidence in the prediction of interdomain orientations, PAE matrices and PTM scores are the ones to use. ipTM scores are used in order to determine the quality of our prediction of protein complexes, and particularly the orientation between different proteins in the complex. All these scores have their appropriate uses as well as their limitations, and these should be kept in mind when choosing which metric to use and how to interpret it. We hope you found this training useful, and thank you for your attention.